tell us.
Well, actually, don't worry about reading the title. I changed it as well. So I was very ambitious in the beginning, but looking at the fact that I have to speak probably already 10 minutes, so I have to restrict myself. I'd like first to thank you, the organizers, for this wonderful meeting. This is actually just my second trip to Cambridge. Last one was last year for only one day to see the architecture. Now I actually enjoy really beautiful spirit of work here. So Blanca introduced very nicely the subject I was going to talk about, and just briefly, again, this is my version of pictures everyone showed before me about reductionism and integration. And briefly, essentially, the model which we all use in translational medicine, especially in pharmacological medicine, is that we come up with a compound, we test it in some sort of cell model, could be cell culture or a computational model, tissue, then some sort of in vivo animal model of a disease. And then suddenly we jump to humans without regard to the fact that, in fact, genetically, physiologically, cellularly, there are so many dimensions of difference between human and various species of mammalian animal models. And a good example is what actually Blanca showed, the Ten-Tuscher and Panfilov model, which probably at the moment the best model we have for human electrophysiology. But in reality, if you carefully look at the currents, overwhelming majority of those currents are coming from guinea pig and dog. Very little is known actually about electrophysiology of human ventriculomyocyte. And of course it's a problem, and perhaps the fact that we don't have too many successes in pharmacological therapy of arrhythmias is exactly for that reason. And we've looked at that problem from a slightly different angle. In the United States, about 2,000 transplants are carried out every year, and according to my computations, probably about 5,000 hearts are not being used for transplantation, despite the fact they are actually donated to transplantation, but for various reasons are not used, age or disease of the donor. And of course, all the explanted hearts from patients are also not used, they are discarded. And of course, all these patients will be happy to donate it to research as well. So we try to take advantage of that and essentially establish a paradigm or establish a platform how to study human heart physiology in freshly isolated human hearts. And because I only have 7 minutes left, so I will restrict myself to only one project, although I would be happy to show later to those who are interested. We've studied so many different parts of human physiology in the past 2 years, including sinoatrial node, atrioventricular node, atrial ventricular arrhythmias. But I will be talking about this particular issue, how transmural dispersion of repolarization and calcium handling changes during failure in humans. So this is a brief summary of what we've done so far in the lab. So we had an opportunity to study 52 human hearts. This is the gender distribution. As you can see, it's primarily male. So we have 1/3 approximately are non-failing hearts. So these are hearts donated to transplantation but not used. As I said again, these are of course not necessarily healthy hearts. This is why they are not used. But mostly it's age. So after 50 years of age, hearts are not used for transplantation in the US. So 1/3 of those hearts is ischemic cardiomyopathy, and remaining 1/3 approximately is various non-ischemic types of cardiomyopathy. So techniques which we are using, and I believe it is really important to apply it to human hearts, including voltage and calcium sensitive dye imaging. So here is an example. We use essentially two CMOS cameras. Tissue is stained by two markers, and you get 10,000 calcium transients and action potentials from the same field of view. Another important part is that numerous studies on gene expression in various animal models of various diseases focus primarily on pore-forming subunits, and that's what also our models primarily talk about. In reality, as you know, there are numerous regulatory subunits. There are also numerous alternative splicing events. So we designed this chip which has almost 9,000 different events from 97 genes. And also another important part to pay attention to is if you take a biopsy or tissue sample, this sample from the heart, this sample contains maybe about 1/3 of actually myocytes. The remaining types of cells are not myocytes. And as a result, the system creates a very high level of noise, which leaves out during measurements very important results. So this study was done actually with selecting carefully 5,000 to 8,000 just myocytes and removing non-myocytes from the sample. So as a result, we've been able to identify almost 70 new genes which are remodeled in humans during heart failure. These are primarily regulatory subunits, not pore-forming subunits, which again opens completely new dimension for modeling efforts in the future. And finally, anatomical distribution of gene expression is very, very important. So again, I don't have time to show details, but we've been able to develop quantitative three-dimensional techniques which can map genes to a specific anatomic structure and show tremendous variability and also importance of variability in gene expression to electrophysiological and mechanical function. So let me first start with a very interesting issue which has been puzzling us for a number of years, which is M cells. So all you are familiar with the issue probably that Charlie Anselevich identified many years ago by showing that in the mid-myocardium of a dog heart there is a subpopulation of cells with extended action potential duration, especially at low heart rate. So we tested the same hypothesis in the human. So we used 8 failing hearts and 5 non-failing human hearts. This is the preparation which we used, which was coronary perfused, LV wedge preparation, which was stained with voltage sensitive dye in this case only. And then we mapped action potentials during endocardial pacing primarily to simulate essentially normal spread of excitation. So these are the hearts we used in a non-failing group. So you can see it's primarily brain damage and also one death from Tylenol overdose. So we found in these 5 hearts essentially 2 types of repolarization. So if you pace in the endocardium, spread of excitation takes about 50 milliseconds, which corresponds quite well to what is known from previous measurements by Dürer. And APD distribution shows tremendous gradient between endocardium and epicardium. And in some hearts, in 2 actually out of those 5, it's fairly steady slope without any prominent peaks and valleys. But in 3 out of 5 hearts, we found a very prominent sharp bump near the subendocardial region of the heart. This bump does not have this morphology as Charlie Anselevich described of a layer, but rather these are really islands of tissue. And in fact, David Rosenbaum showed it previously in a dog heart as well. So if you look carefully at those bumps, you can see, if you draw a line along this dashed line, you can see this distribution of action potential duration. It's really quite prominent, and there is a very sharp gradient of action potential duration versus space. So we introduce a new definition of those M cells as regions which have steepness of this gradient above 15 milliseconds per millimeter. So what happens in the remodeled hearts, in the hearts which are failing? And again, you can see we included in this group actually various types of failure and found very little difference between them. So we have various non-ischemic, ischemic cardiomyopathies, and even one restricted cardiomyopathy, which is exceedingly rare. And our sample was only 1 heart like that. So this is comparison of a previously shown heart on the left and a failing heart on the right. So if you take a heart and pace it at a very, very slow heart rate at 4 seconds cycle length, you can see, of course, this very prominent region. If you accelerate the heart rate to normal 60 beats per minute, you still see this region over here. But in this case, you can see the gradient between the longest and the shortest is reduced tremendously. So essentially, these regions are no longer very, very sharp in the gradient of action potential duration. And if you go to the cycle length of 120 beats per minute, you still see presence of this region, but of course in this case gradients are almost gone and action potential duration is almost uniform across the wall. In the failing heart, interestingly, unlike some of the animal models of heart failure produced by rapid pacing, for example, or pressure overload, we do not see exaggeration or increase in this gradient. So you can see here, this is under very slow heart rate of 4 seconds cycle length, you see almost no prominent gradient. So it's very much leveled, and although you can see at the same time that mid-myocardial cells are indeed those which have slightly longer action potential duration, yet the gradient is much more shallow. At 1 second, this region persists, and 500 milliseconds as well, but again, gradient is reduced even further, of course, with the shortening of action potential duration overall. What is interesting that these regions, although not any more prominent at fast heart rate, they appear to become seeds of alternance and block of conduction at very fast rates. So these are two types of representation of action potential alternance, as APD or as amplitude of action potentials. So we use both definitions because at APD you cannot see it at very fast rate because action potential does not repolarize, let's say, to your defined APD 70 or whatever you take as a definition. So we found that the APD alternants tend to originate in those islands which were previously shown as islands of M cells, if you wish to call them that, although they tend to be subendocardial and not mid-myocardial. So overall, in measurements of APD, you see that in the normal heart there is a prominent gradient from endocardium to epicardium, while in failing hearts this gradient is greatly reduced because of extension of action potential duration in a non-uniform fashion. Endocardial cells extend just a little, mid-myocardial much more, and epicardial extend action potential duration very, very prominently. So as a result, it's all leveled. And you can also see in failing heart very slightly, although non-statistically significant, prominence of action potential duration in the mid-myocardium. So if you take all these numbers together, you see that total transmural gradient in a normal human heart at 30 beats per minute is about 100 milliseconds from endocardium to epicardium, while in the failing heart it's dramatically reduced to 40 milliseconds. At the same time, functional refractory period, of course, is increased because from overall prolongation of action potential duration. So one of the questions we also had, I'm sure as our reviewers, as we just submitted the paper, what about coupling? Maybe these prominent islands of cells are just uncoupled. So we looked at connexin 43 expression and found that in the subepicardial regions and not subendocardial are somewhat poor, have somewhat poor coupling. In failure, it's even more exaggerated. As you can see, in failure, subepicardial layers lose more connexin than mid or endocardial. But overall, coupling is quite good, and we don't see any evidence of uncoupling in those specific regions which we term M cells. So now, what about calcium? Very briefly, in a few minutes I have left, so again, we conducted a separate study. In this case, we stained heart with both voltage and calcium-sensitive dyes, and essentially looked at the same field of view. First of all, about excitation contraction coupling on the upstroke side. So if we look at the activation map and then map of activation constructed from upstrokes of calcium transients, they essentially reflect each other, as you can see, and follow each other faithfully with slight delay. What we found, failing or non-failing in all hearts we've studied, there is a very, very tight coupling between upstroke of action potential and upstroke of calcium transient, on average of 8 milliseconds. So if you look across the field of view, so essentially it's all residing between 7 and 10 milliseconds, being very, very tight. We do not see any prominent delays between voltage and calcium, and we never see calcium preceding voltage, as some studies shown in animal models. So if you look at the reuptake and repolarization, so you see, of course, the same picture as I showed you before in this failing heart. The mid-myocardial layer has somewhat intended to be towards the subendocardium, somewhat longer action potential duration, and it is shown in blue here, statistically significant in the samples. And calcium transient has exactly the same tendency. You have much longer duration of calcium transient, defined at 80% recovery, as compared to epicardial, and it's also strongly statistically significant. But we also looked at the single exponential fit of the tail of reuptake of calcium, and also found that endocardium has a longer time constant as compared to epicardium. So this data has not been yet presented even or published, so we are looking right now at the molecular basis of that, but so far, unfortunately, I don't have anything to report. Hopefully, in a few months, I will be able to. So let me conclude. So the depth of knowledge of the human physiology is significantly less detailed as compared to that of mouse, rat, guinea pig, rabbit, canine, and swine. For example, we still do not have even a single example of recording with microelectrode from human AV node or some other structures. I also believe that functional electrophysiology imaging should guide molecular mapping, such as lipids, RNA, protein, etc., because of significant functional heterogeneity of the human heart. Depending where you take your biopsy from the endocardium, you may end up with very, very different, vastly different density of expression of ion channels or connexins. Left ventricle of non-failing human heart has significant transmural gradient of action potential duration and calcium transient. And there's also evidence from this data of islands of cells with significantly prolonged repolarizations, which could be called M cells, but they are not mid-myocardial. And finally, heart failure prolongs action potential duration and significantly reduces transmural APD gradient in contrast to some animal models. And let me acknowledge, so this work was primarily done by Alexei Glukhov, a postdoc in the lab, and Chin Lo, who did calcium and voltage, but also Vadim Fedorov and Bill Hacker were very involved in this project. Thank you very much.
Thank you very much for it's very amazing data. I mean, we have time for one short question and short answer.
Igor, what is the significance of the different expressions of alpha-actinin throughout the transmural? You showed staining can seem quite different between endo and epi functionally. What is the significance of this difference?
I don't think I showed alpha-actinin. We didn't. No, alpha-actinin was used essentially when you measure connexin 43 expression. If you only have one marker in your field of view, because of differences in your sectioning, you may disrupt the tissue. Essentially, have gaps between cells, which is not because of gaps existing in the intact tissue, but because of your technique. So if you just assess density of connexin 43 expression in the field of view without regard how many myocytes you have in your field of view, then it will be an artifactual. And there are many papers, unfortunately, published like that. What you really need to do, you have to measure how many myocytes you have in the field of view. And that can be done by assessing, for example, alpha-actinin as a marker of myocytes. And then you have to quantify density of connexin 43 relative to density of alpha-actinin.